This is TWIS, This Week in Science, episode number 1032, recorded on Wednesday, October 1st, 2025. This one is for the fearless. Hey, everyone. I'm Dr. Kiki, and tonight on the show, we will fill your head with lizard snakes, swimming cats, and some shocked crabs. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer. The world lost a good one today. This week, science mourns the passing of Dr. Jane Goodall, primatologist and conservationist extraordinaire. For over 6 decades, she blurred the lines between human and chimpanzee, intellect, behavior, and emotion, the first to reveal tool use by non-human hands. She dared to identify emotional states in chimpanzee eyes and kinship in chimpanzee communities. Concepts so foreign to the scientific dogma at the time, and yet so apparent to the young, open-minded observer. She showed us that to understand nature, you must respect it. To save it, you must fight for it. From the jungles of Tanzania to the halls of the United Nations, from bare bones research huts to the Jane Goodall Institute, she leaves behind a scientific movement. We all live in a world forever changed by one woman's courage to listen to the voices of the wild. Forever missed but never forgotten here on This Week in Science, coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki and Blair.
and a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. Before we get into the stories of the week, I want to just continue that incredible disclaimer and our thanking of this amazing woman who really, like you said, changed the way that science was done, the way that we looked at animals. There were researchers.
We kind of take it for granted here on this show because we have a weekly dose of this from Blair, who's been carrying on this movement for us here on the show every week for years. And so to a lot of us, you know, that hearing the early discoveries of Jane Goodall sound like another one-off from Blair. But it was at a time when it was literally nobody believed that chimpanzees or even other animals at all had
individuals. Yeah. And like now, Blair, you can report and we can talk about, duh.
Yeah.
Another study is finding out animals are
never. Yeah.
But as a zoologist and as somebody who has loved animals your whole life, honestly, I know how Jane Goodall impacted me as a female scientist. And good Lord, birds are individuals too. What? Who knew? News at 11. But for yourself, do you have any stories or do you have any thoughts about how important she was to you?
Yeah,
personally?
I remember watching kind of specials on PBS as a kid and her talking about what it was like to be, first of all, to be a woman in a male-dominated field originally, right? But also to do these kind of, you know, to your point in the name of the episode, these fearless things, these things that seemed bonkers to everyone else around her. I'm going to go live with a bunch of chimps. Like, what? But I was lucky enough to meet her once. I used to go to this conference every year called the Wildlife Conservation Network Conference, the Expo in San Francisco. And when I worked at the zoo and I ran teen volunteer programs, I actually would bring teen volunteers with me, and they would table and help out at the event. And then we would always get to go see her keynote at the end of the day as kind of a thank you for volunteering at the event. And hearing her speak would always kind of give me goosebumps and a nice kind of injection of hope, which is something that was always needed in the conservation movement. But gosh darn, lately it's really been needed, right?
Yeah.
But I think the thing that was so impactful to me was actually watching these teen volunteers listen to her, because you would kind of watch, they'd kind of be messing around, maybe messing on their phones, maybe fidgeting, whatever. And then throughout the course of her keynote, they would kind of get more and more interested. And towards the end, you might actually see a couple of them holding hands or hugging each other or really feeling moved by her, because she was so good at like capturing the emotions of a moment and conveying things in a way that seemed matter of fact, but where there was always hope. And I think I will always remember watching these teens listen to her speak at like 80 something years old and inspire a whole nother generation with just, half an hour of her day. It was really special.
And I love that, for her whole life, she has not just, she started with curiosity and a sense of adventure, a spirit of adventure. She wasn't super wealthy. She did have, good family stuff, but she had to buy her own ticket to Africa. And she got a job working for Leakey, but she was more than a secretary, and he gave her this role. And I love the part of the story is that she was supposed to have a chaperone. And so she brought her mom. And that was so the two of them, her and her mom, are out there watching the chimpanzees, trying to feed them and trying to make them at least accept them as parts of the environment and not to be feared. And she was able to make advances that nobody else did before, because she looked at things in a different way. And that is eternally important. And as a woman in science, oh my goodness, I love that she took mom as a chaperone. It's so good. What better chaperone? Mama Bear, you're here. Okay.
Yep.
But then through her life, she wrote about her experience going to one of the conservationist meetings, an annual conference, and really seeing the plight of the chimpanzees, the plight of these animals that were being killed for various reasons. Yay, people, what's up? And she said, oh, I know a lot about these animals. I shouldn't just be focusing on learning about them. I should take what I know and try and make this important to people. And that was the beginning of her activism and her work to really work to create a better world for humans and chimpanzees. Right. So anyway, I don't know how many of you out there have memories of Jane Goodall, or if you do remember Jane Goodall from anything, but a great woman has passed, and her impact is going to last so long after she's gone. It's changed so many things. I love it. We can all make an impact every day. Little things. Yeah. Kindness. Talking about science. We've got an episode of This Week in Science ahead of us right now. Everyone, thanks for joining us. Let's see. I've got science news coming up. Microbes and mouse moms. Old. Analyzed. Junk food memories. Or not. And a lizard snake. What do you have, Justin?
I've got formative fungi. Good news about cancer rates and robot double agents.
Double agents? Agent.
A robot that came in from the cold.
This
Robot
is the
size.
movie at 11. Blair, what's in the animal corner?
Well, I'm very excited to hear about the lizard snake. I'm on pins and needles about that because that just sounds like a legless lizard. I brought swimming jaguars. I brought the resurrection of the dodo and female crabs. They're electric.
Did they do the
Electric
dodo?
lady
Is the dodo back? Or is it news at 11? Oh, God, it's a teaser. I got to
We're
wait
gonna
for
talk about it.
the whole show
Yeah.
to get to the halfway.
If I gotta wait for the lizard snakes, you gotta wait for the dodo. All right?
That's fair. That's fair.
We're all waiting for each other's stories tonight. I'm so excited, and I hope everyone out there is as excited as we are to hear each other talk about science. All right, everybody. This is This Week in Science. If you really enjoy the show and you are not yet subscribed, or you think somebody should be, make sure that you're sharing and you are subscribed and are finding us on your favorite podcast platform. YouTube, Facebook, Twitch, we're there as well. And we have a website, TWIS.org. We are on some social medias. Thank you to Fada for making sure that happens every week. Even when we don't do the show, he helps out with that and makes sure there is social media out there. But yeah, time for the science.
Let's bring it.
Dun, dun, dun. Week ago, 2 weeks ago, whatever, Mars, right? There was the whole like, oh, there's nodules on Mars. Might be life. There have been big questions, though, about other places in the solar system that could harbor life, like the moons of Saturn. A particular moon called Enceladus has been looked at over and over again, considered to possibly have a cold, not water, but maybe like ethanol, ethane, methane kind of sub-ocean beneath the ice, and where there is water as well. And there's due to the tidal and gravitational movement of the moons around Saturn, not that the Enceladus actually has any volcanic activity of its own, but the squishing and moving of the moon just because of the fact that Saturn is so big and is like, come here, and is like grabbing onto Enceladus, and Enceladus is like, no, I'm going to stay in my orbit. And so it's like all squishy. And in the meantime, what happens is the inside of the moon heats up. And so you have the potential for an icy outer shell with a liquid inner ocean. And Enceladus has plumes, these cracks that appear because of the squishy. And so the plumes back years ago, remember the thing called Cassini, that mission?
Oh, yeah, of course.
Went
everywhere.
Dove into Saturn and made me cry. I cried a lot that day. Anyway, Cassini, the data lives on. We have had reports from Cassini for years, people continuing to go through the data. Earlier, Cassini had gotten data from the E ring of the rings of Saturn. And there were these hints. They were like, oh, we found stuff that's organic, these organic molecules, but they've been here in the ring for a while. So these are like half hour to like days old. They were very old, so they could have been irradiated by solar rays, also by the electromagnetic field of Saturn itself. But then there are these plumes. And Cassini actually dove through the plumes of Enceladus at one point, and in doing that, recorded a bunch of stuff that has been finally looked at and reported on. And researchers have just published in Nature Astronomy their analysis of that plume, which has not been sitting out in space in a ring for any period of time. It is just moments old, ejected from that watery interior under the ice shelf.
Squishy core.
Yeah, squishy. Yeah. Anyway, my sound effects are not great tonight. Nature Astronomy.
It's the beginning of the show. There's time. You could turn it around.
I can turn the sound effects around for sure. Nature Astronomy, the paper published today, detection of organic compounds from freshly ejected ice grains from Enceladus's ocean. They have using the cosmic dust analyzer spectra revealed previously unobserved molecular fragments identifying aliphatic, heterocyclic ester alkenes, ethers, ethyls, and tentatively nitrogen and oxygen bearing compounds, according to the abstract. So finally, we have the potential for organic molecules that could, especially with the nitrogen, the amine, ethyl, all this stuff, maybe there could be stuff, microbes, living in that watery.
But that's
So
just
that's like-
chemistry.
That's like the equivalent of like scooping out ocean water and saying, there's some environmental DNA. I didn't get a fish, but I got, some fish juice in the
chemistry.
ocean. I'm saying it's analogous. It's not the
Juice in
same.
the ocean.
I'm not saying that you got DNA from the bacteria, but I'm saying like that's basically what you're doing, right? Is you're getting an artifact of something that could indicate that there's life under there because you got this point.
And yeah, but I mean, if you had a signal of fish juice and you didn't know that there were fish,
Yeah.
but you had that signal, you'd be like, oh,
Didn't know what a fish was.
fish juice. There's something fishy about this, right? Yeah.
Mm-hmm.
I'm
What you
sorry.
have is just chemistry, as far as I'm concerned.
But the ice grains that the data, these spectral analyses have identified, these ethyl compounds, different compounds that are relevant for organic chemistry, right? Which is potentially the substrate for biochemistry, which would be the chemistry of life. And they found some very, very solid signals. And in this wonderful article, they actually put forward a flowchart of potential chemical pathways between organic compounds on Enceladus. So to suggest that
Any of them include a fish?
no fish, no. But they do
No DNA.
include these esters, ethyls, alkenes, all the things that they
I remember those
identified
words
and
from
how they
chemistry.
fit, right?
Exactly. They're
Yeah.
chemistry words.
Metabolism and
Yeah.
proteins and all sorts of stuff. Yeah.
Wait, proteins? They found proteins?
No. Oh my gosh. I'm
Oh,
sorry I
thank
said
you.
that.
Yeah. Easy. Chemistry.
Chemical pathways that are associated with life activities. And they suggest a whole bunch of chemical pathways between the organic compounds to suggest how they could have arisen, how they could interact with each other, and how they could be in existence in this liquid ocean under the crust of ice. Not that they have proven it in any way. This is just they found some stuff in an ice plume that Cassini flew through a while back, and
But we're still talking about chemistry interactions that can explain the chemistry that they found.
Mm-hmm.
yes,
And if they
Yep.
can explain it without needing life,
Mm-hmm.
which is, I think, what they've done, is more of a sign that there's not life.
it is these are pathways. These are potential pathways. They have no idea, but they could be significant in the context of a substrate or like we're not saying life. We're not like the dude on the Ancient Aliens show.
It's the same as a couple weeks ago. We're saying there's a chance.
This is
No, hang on. No,
But
let me
this-
slow
actually,
it down,
this
though,
is-
because
so I'm
maybe
gonna go-
I'm
I'm gonna
being
go
slow.
backwards.
I might be
I'm
being
gonna go-
slow because I'm missing
But
something.
no, a couple of weeks ago, Justin was like, but this is a really like the chemistry and the way they put it forward is that they're really, really positive. And they were using scientist speak to like, kind of couch it in terms that couldn't be taken to be certainty. And I still think, we need to look at it, but they had much stronger signal and like the chemistry is more suggestive of the recent Mars finding than this chemistry is. This is just organic molecule fragments. So
I think it's an important distinction. I appreciate that because I think that there's a chance, but it's like magnitudes different. It's just so
Yeah,
like,
yeah. The Mars one is
there's a
much
non-zero
bigger hit.
chance, but this is a much smaller non-zero chance. Just like the stuff is there, and Mars is like, it looks halfway made.
Yeah. So that's the different, like that is the kind of counterpoint, right? So this particular study, they're finding evidence, and the more evidence they have, the more, I guess, like support they have scientifically to send more missions. So the European Space Agency wants to send a mission to Enceladus to actually go land something and see if it can crack through the ice and, take a really good look at what's there. We want to go and find the possibilities in our solar system. Where else could life be found?
have one, without going too far on a tangent. Oops, too late. I don't understand. Please explain to me, because the moon,
No.
our moon does not rotate, right? You're always kind of, it's doing this.
Well,
right?
it depends on where you're standing.
Yeah, it kind of does.
It rotates, but
it does like one rotation in the time period of a 24-hour orbit.
Yeah.
Yeah.
So the same side of it is
offset
always
by its
facing
orbit.
Earth.
Yes.
Exactly.
That's what I'm saying. So if we are assuming the same is true for this moon,
I would.
I have trouble understanding how you can have a frozen surface and a
got cracks.
squishy core.
Right, because it's so big and the gravitational forces at play, like, the gravitational forces of the moon and our orbit around the sun influence the tides on our planet, right? And so there are massive forces at play that move things.
Yes.
Those forces are also influencing the rock and, but to a lesser extent because of the size of the bodies and the forces at play. There are interplay based on how far away the moon is, where it is, on what side of the Earth at what time. And so there is a constant, pull. The two bodies are pulling on each other. And so Saturn is massive. and Enceladus is in this spot, a sweet spot, where it's not being pulled in to crash into Saturn like Cassini, but its orbit and the place it holds is being tugged on by that massive gravitational field. And so this really
and
cool...
so the tug is enough to like keep the juices moving.
Yeah.
Okay, got it. Okay.
Yep.
That makes sense.
Yep.
Interesting.
It's enough to heat up the interior of that body.
Yeah.
Enough. Just enough. But we're talking about not frozen, not water. We're talking about like methane, right? Liquid methane is a much, much colder freezing point. So, the scales are different, but it's like what, that's why they look at extremophiles, right? Go to Antarctica, why they go to different places to kind of see like, okay, how can things live in really cold, really acidic, really hot environments?
The extremophiles came to us from out. They started on Enceladus. They came over, the extremophiles, panspermia, right? So there you go.
Nah, they started on Atlas 3, whatever, no. Interstellar interlopers brought them.
Yeah.
Anyway, that's a fun story.
Well, the fun thing is that chemistry is already everywhere. We don't have to panspermia it at all. And we're getting more and more signs, the more places that we look, that chemistry exists like we might expect it to everywhere in the universe.
Yeah.
Life on Earth did not start everywhere all at once. In the beginning, there was just chemistry sloshing about in our oceans.
Yeah.
From this chemistry, single-celled life emerged, proliferated, and dominated as life's only representative. Right? So come to Earth, you sample the ocean, there's no fish here, but there were single-celled life forms dominating planet Earth. So then some revolutionary change occurred, not all at once, but many times, more than 5 separate times. There was, as a complex multicellular life evolved independently in what we have classified or categorized as 5 major groups. We've got animals, land plants, fungi, red algae, and brown algae. These are the basic 5 major life groups on planet Earth. 5 categories of organisms, complex organisms with different origins and different timelines. Each one, cells took on specialized jobs and were organized into distinct organ and tissue systems. This evolutionary leap required sophisticated new genomic tools for each line, including mechanisms for cells to adhere to one another, to communicate across the larger organism, and each of those arose independently of all the other major groups. So, dating the order of these evolutions relies heavily on our fossil record, which is science's geological calendar for things that happened a really long time ago. Red algae shows up about 1.6 billion years ago in a candidate seaweed-like fossil from India. Animals don't show up until like a billion years later, and are around 600 million years ago in our oldest recorded relative, which is known as the quilted pancake.
It's a sponge.
Excuse me?
The ancestor of all mankind and animal kingdom earliest representative was a quilted pancake.
That's the
Land
name
plant.
of the sponge? I love it. What?
Oh, it's something Dickinsonia or something like that. It's I think a literature fan maybe got to name it for the official name, but it's also known as the quilted pancake. Land plants took root about 470 million years ago, and we know we've got that based on fossil spores. So, yeah, maybe older, but that's our earliest time point. Brown algae, this is like kelp, stuff in the ocean that we're kind of used to the plants or the fish eating, diversified tens to hundreds of millions of years after plants. So we have there our current chronological picture of life's emerging complexity, with one notable exception: fungi. The fungi kingdom's history is a riddle wrapped in a mystery, stuffed inside an enigma, long thought to be lost to the geological records deleted spam folder. Basically, it's their soft.
It's because they're so soft and
soft
squishy.
bodies. Yeah, right.
They don't
we have
fossilize
any
very well.
fossils. Yeah.
Yeah. So unlike animals or plants, which appear to have a single origin to our complex multicellularity, we also know that fungi seem to actually have evolved that revolutionary multicellularity multiple times. Diverse array of unicellular ancestors to fungi. So fungi as a group is also all of these other first-time multicellular life forms. So we've kind of talked before about how, sort of life seems to have only emerged from one distinct time point, and still we're still sort of there at the unicellular level. We don't have necessarily a diversity of unicellular life time pointed out, but multicellular life started multiple times that we know of, and fungi could have kept going without the rest of us, and we would have had continued diversity of just fungi. So part of this is like also making it difficult to pinpoint a single origin for fungi, is that not all fungi began at the same time point. So when we talk about the 5 emergences, we're missing origins of this group by wide margins. To overcome this, scientists are now focused on the molecular clock, the idea that genetic mutations accumulate in an organism's DNA at a relatively steady rate over time, over generations, kind of like a ticks of a clock. But it's not calibrated, meaning it can't really give us years. It can tell us sort of generationally, but that doesn't always answer everything, right? So they need anchor points somewhere in there to say, okay, how does this relate to this, and where is that in the fossil record, and is that a given point? And there's a great scarcity of the fossils and fungal, so that's also a challenge. So horizontal gene transfer is what they've now turned to as a form of a fossil record. So the way they've got this constructed is, if you have genes from lineage A is found to have jumped into lineage B, you can suggest that lineage A is at least as old and older than lineage B. Now, lineage B could also be very old, and it just got it, but we're at least knowing that they overlapped at this time point. And then
they were swapping genes at this point in time.
Right. So they were both coexisted at this set point in time somewhere. Okay. So by identifying 17 of these transfers, and this is only going to be in currently extant or currently living fungal organisms, right? So this can't tell us all the fungal forms that died off before the ones that are living, if there were any. But we got this sort of older than, younger than relationship now in the fossil record with 17 of these time point stamps. And the analysis suggests a common ancestor for living fungi dating to roughly 1.4 to 0.9 billion years ago. So that's well before the 470 million year old age of land plants.
That's a long time ago.
And what's super interesting about that is, of course, the fungi are land creatures, which means that for maybe a billion or more than a billion years before plants took root on the land, the fungi were working away. Their ancestors were working away out there.
Preparing the soil.
Exactly. Recycling nutrients, altering land chemistry, perhaps changing the atmosphere, partnering.
Do you know for sure these are terrestrial fungi, Justin?
Yes.
Okay. Interesting.
Yes. The paper couches them as ecosystem engineers, creating the first primitive soils, fundamentally altering the terrestrial environment. So,
are you talking, Blair? Are you asking if there are there aquatic fungi?
There are, yeah.
Hot chew, says Kiki.
Tried to turn.
Yeah, there are underwater fungi. That is a thing.
Oh, no doubt. But what is their origin? I don't know.
Right. So yeah, that's what I was curious about. Is do we know for sure that terrestrial fungi came first?
The genes that are present, maybe, to deal with aquatic environments versus atmospheric and soil. I have no idea. I'm making something up right now, but that's my hypothesis.
That I don't know. I didn't know about underwater fungi.
Yeah.
So, but possibly a billion more years of fungal recycling of nutrients on land, creating these soils. So, and then in this view, then plants, when they get to land, they aren't so much forging a new path for life as they're jumping into an ecosystem that has already got had eons of ancient and persistent activity by the fungal friends preparing the way.
And now I want the cartoon to be called Fungal Friends. Saturday morning science cartoon.
Yeah.
This is my fungal friend, Basidiomycetes. That taking it back that long, that fungi were prevalent and active I mean, it makes sense that there would be something complex and working before
Yeah.
the plants.
Well, we know that the
land,
planet
but it-
was, and I forgot what the
Yeah.
name of it is, but those weird tree-like forms
Mhm.
that were like giant, 30-meter
Yes.
tall
Yep.
or 10-meter tall, whatever it was.
That was just like last month we talked about those.
Mhm.
Yeah, those weird fungal trees that they say isn't exactly related to any current day fungus, but now, and under this light of understanding that the fungal community originated from different single cell organisms multiple times, it doesn't need to be related to current fungi to be a fungi.
Right.
By our broad definition.
I love this new technique, though, of using the horizontal gene transfer as a way to to control for the fact that we don't know, we don't have fossilized stuff. We can't go back and do the radio dating in the traditional way. And if this one was doing this and this was doing this and the same genes are at the same time. Oh my gosh.
I just love that it's not any fancy math. It's just like, we know this was happening at this time, and we know that this was happening at the same time as this thing.
Mm-hmm.
So basically, we're just using the transverse property of like, if A equals B and B equals C, then A equals C. So like,
Well, yeah. Wait, don't take my cartoon version of how they did it.
No, that's how relative dating works. That is 100% how relative dating works. Is you're just saying like, I know it's at least this old because it's associated with this other thing. Like that's how like relative dating works when you go like, I know this fossil is here and this fossil is from this time and this fossil's below that fossil, so it's older than that. Like I love that it's not this crazy calculus that's happening. It's just like it's all relative logic. I think it's beautiful. And I love how like simple the math is. It's I've always thought it was really cool that it's like there's no trick. That's it. That's the whole, you're just using basic logic. Love it.
And then some math. There was math. But
Yeah, of course there was math when you were doing the actual radioactive dating and other things. But in terms of the relative dating, there's not really any math. It's just, this is more than that.
I love it. But this is brilliant. It makes so much sense. I love this this new way of measuring, and maybe it'll come up with some new discoveries of other microbes, other organisms that are soft-bodied that we hadn't hit on yet. Because horizontal gene transfer is not just like it's a lot.
But like a soft-bodied, the continents all being dominated by soft-bodied life. Like, can we even imagine the fungal terrestrial scene without any plants, animals, or insects or anything, where the world for all the life on land is just varieties of fungi? Like, wow. That world existed, and maybe for 1 billion years.
Rock years.
That's wild.
Blair. No, we debate every once in a while about whether or not fungi are animals, but did you want to talk about animals?
Yeah.
Yeah.
Cats. Is your understanding that they like water or they do not like water?
No choice.
People say they don't like water. You can if you get them in water when they're babies, you can get them to like it. They can take baths and stuff. Yeah, no, cats are not like big. People don't think cats are big swimmers.
Yeah.
Right.
So when I worked at the zoo, we would say most species of cats avoid the water. There's a bunch of reasons for that. There's a lot of cats that live in cold climates, so it would be a bad evolutionary strategy to get wet when it's really cold. A lot of cats do not have webbed feet, so they are poor swimmers. But tigers, the two types of
I love that you just said
a
lot of cats do not have webbed feet. I don't think people would think that about cats very often.
Yeah. Tigers have webbed feet. Fishing cats have webbed feet. They're good swimmers. Jaguars, which is what this study is about that I brought here. Jaguars have been understood to be able to swim a maximum of about 200 meters. So they don't mind getting wet. They don't mind jumping in water to grab a meal or to cool off.
That is
but
much
there's
further than I
not-
think I can swim. 200 meters?
You can I could definitely
Without,
swim 200 meters.
200 meters?
Yes.
That's
two lengths of a pool, right? Is it right? An Olympic pool?
It's like two football fields.
Yeah, I think
That's a long way,
an Olympic
Blair.
pool. Yeah, it'd be fine.
Anyway,
But point being, yes, it's farther than a lot of people would imagine a cat swimming, but you're not swimming long distances. And so, for example, in jaguar conservation, it has long been assumed that reservoirs, wider stretches of river, and other bodies of water are a barrier to movement. However, this week, a jaguar has been identified based on their unique spot patterns on the mainland in Brazil, in Goiás state, and then also on an island 2.48 kilometers away. They were both recorded by a camera trap.
Kilometers.
Three times this cat was recorded on the mainland, and then one time it was recorded on the island. They are very confident this is the same cat. By looking at all of the potential trajectories, I love this, that they're not like, well, maybe he didn't just swim straight. Like, if you look at any of the possible paths to get from the mainland to this island, either this individual cat swam about 2.5 kilometers straight, or, more likely, they used the stepping stone option. So there's a little islet that was about 1 kilometer away, and then there was a 1.3 kilometer swim to get to the island from there. So they were able to kind of take a break and then keep going. So kind of like the relative dating, based on the previous assumption that they can't swim more than 200 meters, would it make more sense that they swam 2.5 kilometers or that they swam 1 and then 1.3, right? So the assumption would be that they stopped halfway. All that being said, though, even if they could swim a maximum in that case of 1.3 kilometers, that is a huge difference from 200 meters. So this completely changes jaguar conservation. This completely changes what they would consider, quote unquote, an absolute barrier for a large carnivore. This impacts understanding of island biogeography and why certain islands don't have large predators. Like, potentially there's something else going on here. But I think the thing that's really interesting is they bring up a new idea of an aquatic cost scale for modeling connectivity between land masses. So you look at a low, medium, and a high cost ranking for water crossings. So yes, mostly you might not see jaguars swim more than like 200 meters because that's what they're comfortable doing. So low cost they would identify as less than 300 meters, medium between 300 and 1 kilometer, and then higher would be a kilometer or larger of just open water. And so from that, they're recognizing low, medium, and high difficulty. And so what is the reason to be moving, right? Are you fleeing humans? Are you going after a high quality food source? Are you going after mates? Are there things pushing you out of the space that you're in? Did your habitat get fragmented, right?
Yeah. Are you into extreme sports?
Yes. Are you a jaguar who just loves to swim?
Wants to swim
for the Olympics.
Right? Again, yeah. I know that there's humans who could make this swim. I would maybe struggle at the 200 meters. That's a long way for me to swim.
For
you to swim.
Right. But this is my point.
Swimming a kilometer, that's a lot.
Is there as much diversity in like jaguar athleticism as there is in humans? Like, there's human athletes who are going to outperform me at every athletic task, right?
There's like an easy answer to that question, and there's a hard answer to that question. Okay. The easy answer is No, there's not as much variability. And that's because there's evolutionary pressure. The harder answer to that, the more complicated answer is that because of habitat fragmentation, because of human encroachment, because of everything else going on, there are actually more kind of like microcosms in habitats than there ever have been before. And so you might find some specialization happening. This might be a recent selective pressure that has been put on jaguars because of human activity. There's a lot of things that could go into this. Like maybe jaguars could only swim 200 meters until 20 years ago. And then the last 3 generations of jaguars have had more pressure on them, right?
Or maybe just this jaguar.
Just this one. Yeah.
It's turns out I'm a great swimmer. I'm going to go where I want.
It's like the garbage dump seagull, right? It's like the gull who ended up at the compost pile in the Central Valley and was like, "Oh, this truck is amazing."
Yeah.
She did that a few times. She's amazing, but how many other birds did?
Right.
But so,
or did they have a whale friend?
Yeah, individuals can be exceptional, right? But there's also a question of the metabolic needs of these different cats, right? So we have cheetahs who can run super, super fast for a little bit and then they get super tired out. They're like made to run, right? And then you have like tigers, you said they have webbed feet, like they are going to get in the water and they'll swim. And they're water cats and they'll be fine with it. And they go after aquatic prey. But so what are the lifestyles of these different animals? I know that I have heard about jaguars in the jungles where there are rivers being able to catch prey along the riverbank or get in the water for a short period of time to catch prey. And then they take it back up to a tree and they like hang their kill up in a tree where they eat it. But so I'm just wondering about like the different metabolic needs of the cats and like why would they say only 200 meters? Is it just that they never saw it or that they are thinking that metabolically it would be unlikely?
So this is of course reminding me of working in the zoo field and designing exhibits based on what is understood that an animal can and cannot do. And how much that also has changed over the last even 20 years that I've been in the field. Yeah. And so
it seems like every few years,
by people inspired by Jane.
Well, that and every few years they realize, oh, tigers can jump how high? Oh gosh, we need to build an even bigger fence next time.
Yeah, or, chimps hate water. Oh, we just discovered there was a chimp standing in open water beckoning people to come in to the oh my
Play
God.
With me.
But I think that's my point is like it's so easy to look at animal adaptations in a vacuum, right? And forget that we are on the treadmill. It's moving. So even if 50 years ago or 200 years ago, when some white guy showed up in Brazil and wrote about jaguars and decided that this is what jaguars could do, that might actually have been the case 200 years ago. But
or
also,
they didn't watch very far because they were comfortable in their little camp, right? And they didn't see all the things.
It could have been either. It could have been that the observation was flawed and it has not been challenged, even though it should have been. It also could be, and it can be both, right? That adaptations continue. And especially remember, like we live, the human lifespan is what, like 60 to 100 years, just being like really, really wide, right? But
imagine a jaguar, what, like 20 or 30? Maybe like if
no,
40?
like 12, like 8 to 15.
That's 20-ish.
And so
okay.
yeah. But I'm saying like on average is probably closer to 10, right? And so that's a lot of generations.
So like a large dog. Got it.
Right. And so that means that there's an opportunity for a lot of adaptation over that time, especially with humans encroaching, with new prey, with livestock and farming, with all of these things happening, there is a lot of opportunity for adaptation and evolution. And we are like on the treadmill, like it's happening. Like we are watching animals evolve around us. And we forget that, I think, all the time, that we kind of have this idea because we're at this little pinprick in evolutionary history that
everything is the same. Everything is only 6,000 years old. It's all the same and has never changed and will never change.
Yes, no. And I will say
I did
a quick Google,
accurate, and I apologize.
Justin.
I apologize for backing there for a second.
No. An Olympic-sized pool is 50 meters long, so that is 4 laps.
Done.
In an Olympic pool.
Pull me out.
Yeah. Well, I'm just saying I could do it, so it'd be fine.
Like you say, like I can do it, it'd be fine. Like it would be a walk in the park. You'd be like,
yes. I mean, me today, you're correct, but 5 years ago, it would have been no problem at all. I wouldn't even have been short of breath. No.
Oh gosh.
But anyway,
this is exciting. Yeah. So big carnivores like the jaguar.
Yeah.
Could have expanded their ranges and ended up in different places than in different ways than we previously thought. Where previously we considered it all to be overland or whatever. Now there are explanations that can be used.
Mm-hmm. Yeah, but we need more observations. One jaguar does not a pattern make, right?
No, it could just be the, extreme swimmer jaguar, but yeah. That is so cool. I love, though, the more that we are tracking stuff, the more we're able to come up with these kinds of, data points. Yeah. Be able to see that horizontal gene transfer in the jaguars. What about mice? We don't look at them enough, do we? Ever? No. Did you know? No, I'm not going to go there. Anyway, mice can be used as a model species for a lot of studies, including maternity. And so it's easy to manipulate the mice because thanks to places like the Jackson Lab, we can have germ-free mice and we can have wild-type mice that have all of the germs, microbes. So we've had conversations previously about the concern of offspring. So when babies are being born, what do C-sections do versus natural birth? How do you control for the microbes and the immune system of the child? But this study looked at how the immune system and microbes influenced the eggs of the mother. So they took germ-free mice, and so those that had no microbes, and regular mice, and they set them up in a few different situations. And they did a number of different manipulations to get to the results of this study, which really showed the bottom line is microbes influence the immune system and lead to a bunch of short-chain fatty acids and other molecules. So acetate, propionate, butyrate. These compounds make the ovaries and the oocytes in mice. They haven't looked at it in people yet, I'm just going to say, but better. So the germ-free mice showed oocyte depletion. They showed, and they looked at it at different stages of development. So like the first state when the oocytes were starting to form, at different points of weaning from their parent, from their mother, and then on to becoming an adult and having their full complement of oocytes and being reproductively active. They were able to show through the manipulations of this study that there are all sorts of points through the mouse life cycle at which microbes influence these molecules in the gut, really. So this is like not in the follicles, this is in the gut. And those molecules end up influencing the immune system and ovarian homeostasis and the follicle reserve. And so microbes make for better fertility in mice.
So this isn't a guy. Is this essentially? I mean, it's a way of telling the story, and it's interesting to see the downstream effects. But this is, I mean, if you just had a malnourished mouse, I mean, isn't this essentially saying
that's the malnourishment is also going to have impacts that will deplete the follicle reserve because the body is going to do what it can to stay alive, right?
Right, because this is essentially saying that microbes are important for, like, the way I'm hearing it anyway, the way it's taking me is like microbes are essential for nutrients, breaking down food to pull the nutrients out, which then affects XYZ. But like if you just had a malnourished mouse, you would see. So I mean, it's, to me, they're connecting dots, which are real.
No, I get what you're saying here. So they did try and feed mice different fat additive diets to be able to make up for the lack of microbes and other things, but the microbes were the key. That there are potentially ways to, manipulate it, but it's not dietary.
But it's
microbially produced at the scene. This is just in one of the first studies that looked at it in this depth, and it's just
I get it, but isn't like the whole point of the gut microbiome is that it is extracting nutrients from those raw ingredients that we put into the gut. So whether you've got all the nutrients in the food, whatever it is that you're eating, if you don't have the microbes to break it down to utilize it and then create whatever they're creating as well, so it's, again, to me, it's just they've malnourished.
But I feel like we've heard other connections between the microbiome and other systems in the body, right? Like I'm not making it up that the microbiome has previously been linked to hormone regulation, right? Like we've talked about that, I do believe.
So the lining of it all is neuronal tissue. So it is connected also to
the gut, your brain,
the entire nervous system.
Yes, absolutely. But I'm saying if we know that the microbiome can have an impact on hormones, then naturally that would impact your follicles and your follicle development.
Yes.
So that's also, it's not, it can't just be the nutrients. It feels like the microbiome is doing so many other.
So the microbiome, the colonization of, these short chain fatty acids were signals that they were able to measure. So they were able to see the dysfunction slightly alleviated in germ-free mice when they gave them the short chain fatty acids, but it wasn't to the same level as mice that had never, that had microbes. The microbes themselves aren't just making these compounds; they're also they're influencing signaling, and so the host microbe interactions it's an environment, right? An ecosystem, and you can't just go and stick. You go and you can stick the food in it, right? Make it not starved, but it's not the same as the ecosystem effects of what's going on with the back and forth between the microbes and the mother. And so, of course, this is not, the final news on anything. This is published in Cell Host and Microbe. These researchers were able to just show that the gut microbiota impacted reproductive capacity, and that the mice that did not have microbes, they saw their reproductive capacity fail more quickly and faster than mice that had conventional, or that were, that had microbes. And so the question is, right.
I would still push for, this is because the nervous system, the body brain, believes it's starving because it's not getting the signal feedback that it would expect from a microbiome that is doing the work of nutrients. Even if you've supplied the nutrients may not communicate on their own to say, hey, I'm here, everything's fine. You have this entire community that's sending out signals of these processes that are going on, that are informing the neuronal gut tissue.
And they're saying, yeah, this is correlation. We are seeing that the microbe, there is a no microbe difference, and these short chain fatty acids are involved. That's going on there. And they also found the thing that I think is so fascinating at the different time points in development, the shifts in the diet, they match the shift in the ovary and the follicle development matches the shifts in development. So changing from like suckling to being weaned and eating solid food. So they said as they looked at these transitions, they saw the weaning transition was marked by the emergence of Clostridia members. This transition coincided with the onset of solid food intake, which introduces dietary polysaccharides and complex carbohydrates to the intestine that support bacterial fermentation and production of microbial metabolites. And at that point, there is also a shift in the development of the follicles, the ovarian follicles. Additionally, at weaning, there on day 21 to 28, you have different bacteria emerging and diversifying as changes occur in the follicle, which I think the matching of development and the emergence of certain bacterial species that would be bringing certain short-chain fatty acids with their metabolic processes, that it turns it into a really interesting question. And of course, we have no idea if this is what happens in people. No clue. But we do know that there are lots of people who have fertility issues. And if it has something to do with diet, if there are targets that can be addressed, that maybe there is something that can be done that's, maybe an easy intervention as opposed to something that is much more complex. People spend tens of thousands of dollars on and stress out, like their lives are turned upside down by an IVF. And if that didn't have to be the process, for
some
more
poop
people.
pills instead.
Exactly, right? So,
Dr. Justin, not a real doctor, poop pills for whatever ails you. It's really not at this point an illogical thing to, no matter what ails you, go and have your go in now and get a baseline, and then go in if you're feeling ill or having something that you're trying to address, and see if it's changed, or see how different you are from the wild type human population, whatever.
Yeah, and with these mouse experiments, they're mice in cages, and this is so very controlled, but they gave them fiber, and it changed their fertility. So, there is certain aspects to this that it's like, what? Like that, they're very, very simple interventions, and they saw impacts on, on the oocyte maturation. So anyway, mice, not people, but they've never done like this. It's such an there's so much to be dug into here. And I think you're exactly right, Justin. Like there are other explanations possible, right? But it needs.
Well, it's a combination of I think yeah. There's definitely though a cell. I would suggest in signaling to the central nervous system into the brain about whether or not it's cost effective to be doing reproduction right now, based on what the gut is telling. The gut is going to be basically like the eyes, ears, and sense of touch for how the organism is doing for the brain to make all of these complex decisions.
Yeah.
And it's all going to be signaling that seems to be happening in the gut from activity of microbiome. And this is a great example of that, really. I mean, it's really like very cut and dry. Here's how your body functions with, and here's how your body functions without input from your microbial guests. Or crew, not guests. I guess they're the crew of the ship, not really the guests.
And what I think is interesting, they had these mice on high-fat diets, which when you think of like the Western diet and what we eat, low fiber, high fat, our reproduction rates are going, our fertility rates are dropping. And then they gave them fiber, and the mice saw returns of their fertility. So anyway, or a decrease in the loss of their fertility. Not people, mice. Mice in labs, very controlled. But it's a really great question.
I can run through it really quick. In the segment that was long abandoned and then brought back just for this occasion, just good news, everybody! Cancer. Okay. I'll recap really quickly the story that nobody could hear. In the last 30 years, cancer mortality has fallen in half. At the same time, mortality remained unchanged at 5.9 deaths per 100,000 in the last 30 years, because while mortality has fallen in half, it's largely because detection of new cancer has doubled. So incidents of cancer being recorded have doubled. Actual mortality per 100,000 has not moved.
So the same number of people are dying relative to population,
right?
But cancer is less deadly. So how many people it's less deadly in that, like, the number of cases relative to the number of deaths is different, right? But how many people were diagnosed with cancer via autopsy, right? Like, how many people were diagnosed with cancer when they were already dead?
Oh no, that was probably, that's a great point. That was probably much more.
They cut open and saw tumors everywhere, right? Like, that's part of the detection.
So I'm still trying to figure out. So in this study, they have suggested that the higher incidence of cancer rates that we've been seeing through diagnoses are because diagnoses are way up. That we're detecting more types of cancer earlier, and that it has, it's not that there's a new cancer epidemic hitting, that it's the same cancers we've always had, same basically rates, I guess. It's just that our ability to detect them and detect them much earlier has improved. Okay. But then I have to question if it's, what is it, 5.9, did I say? Deaths per 100,000 is 5.9 deaths per 100,000 is unchanged between 1992 and 2022. That tells me we've gotten no better at fighting cancer.
Interesting.
There are more cases. We're better at finding it and seeing it.
There's double the cases because of diagnostics.
But if there are double the cases, we're also better at treating it because we are keeping up with and continuing the decrease in.
You would then
deaths,
think
right?
that the deaths per 100,000 would go down. Now, the thing that Blair said, how many of these are via autopsy, is interesting, because that throws in the possibility that we just missed causes of death previously.
Sure.
It's still, I mean, then you could say like, yes, actually the real number from 1992 should have been more like 10 per 100,000, but because we weren't so as good at diagnosing, we missed a lot of things that were cancer that killed people. But the number's unchanged, which is
there's another possibility.
Coincident.
It would be very coincidental, but it is possible that people are getting cancer more because of chemicals and PFAS and, right. Whatever. Just name random things.
For sure.
High fat diets.
But that we are better at treating it. And the rate of increased cancer and the rate of treatment is like canceling each other out.
Exactly. No, we have to be better at treating it to be able to keep up with the wave of new diagnoses.
This current study, which is out of Harvard Medical School, Brigham and Women's Hospital, and Dell Medical School researchers are saying no, the increased rates of cancer, the doubling of the rates of cancer in the last 30 years is wholly due to improved screening and earlier detection.
Which is amazing, because even though the same number of people are dying, people are probably in less pain.
So there's another study out this week that is suggesting that the increase in cancer rates, colon cancer specifically, in younger adults is specifically related to diet. And they're trying to tie it to highly processed foods, which could be possible. There could be things, but we have this one study that came out and saying a lot more younger people are getting cancer. But what you're saying,
and it actually points out
diagnosing it more.
It actually here it states that colorectal mortality rose slightly, while its incidence rose much faster. So not everything moved together. Thyroid and kidney cancers display sharp incidence increases alongside stable or declining mortality patterns. So that's more consistent with potential overdiagnosis, finding lots of what you would call non-fatal or maybe non-dangerous cancers.
Mm-hmm.
Cancer happens all the time. It's not all it gets to the point where it's actually life-threatening. It's a lot of things, sort of slides moving in different ways. The part that still is bothering me is that if it was like, it was 5.9 and now it's 5.5, or if there was any difference in that mortality rate, I'd be like, okay, yeah, it's a bunch of different things at play. But looking at that mortality rate, I'm like, are we just not any better at this? That's the number of people per 100,000 who die of cancer. So
we are.
It could be a coincidence, but it bothers me that it's the same number.
But it's over many years that we have insurance companies here in the United States paying for not treatment, but detection, right? So we have more testing, we have better testing. People are actually going to the doctor more, especially since the America Cares Act. People have insurance, people are going, they're getting tested. You have cancer, right? As opposed to just dying and not knowing why, right? So now we have better record keeping.
We have better record keeping. And that's actually what's disturbing me, because that's saying even though we've doubled our detection rate, our mortality rate hasn't really changed. The only reason it's down 50%, which is sounds like we're winning, is because we've doubled the rate of the number of people who we've diagnosed with cancer.
Yeah.
Well, the actual number per 100,000 hasn't moved. That's frightening. That's terrifying.
Well,
that's not good news at all.
I thought you said it was down by 50%. That's good.
But that's because the diagnosis
Percentages.
It's
I
up,
know.
doubled.
Numbers. I know. Yeah.
Anyway, yeah.
Yeah.
So but it may just be a
That's
giant
good
coincidence. Maybe it's not.
I think so. I don't think so.
How come none of my good news stories ever end in good news? It started great, cancer's down 50%.
Because you pick them for that
Yeah, I
reason.
was gonna say, Justin, you have all the power
Yeah.
here.
No, the problem is I never read the dang story to the end. I'm like, oh, this looks good. I'll put it in there. It's good news.
So
And then
you're
I read
learning about
live
it
on-
with all of
Live
on
us.
the
I
air.
see. Yeah.
Lies,
Mhm.
lies, and damn lies. Oh my gosh. Well, I got a lizard.
Yeah, let's hear about this lizard
Let's hear
snake.
more
Give
about
me
this.
it.
Yeah. Okay,
Legless
so
lizard.
well, it has legs. It's not a snake.
So then it's a lizard.
It's a lizard.
Lizard.
Wait a second.
But maybe it is
This
like
whole
an early
talk about
ancestor.
lies, damn lies, and more. Whatever.
I mean, it would not be as exciting for me to say, I've got a story about a fossil squamate.
Yeah, it would be exciting to me.
Okay, back up for those of us who don't know what a squamate
All right.
is.
Squamates, lizards and snakes. Lots of them around the world. They maybe originated about 190 million years ago, much younger than those fungi you were talking about. Researchers have found a new fossil, but it's old, in Scotland, and they have called it Brugnathar elgolensis because it was found in Scotland, and so that's what you do. They have taken Brugnathar and compared it based on its morphology and other aspects that they have been able to measure with in-group and out-group phylogenetic comparison. And they're like, oh, okay, it's definitely a lizard, maybe early snake. It's very similar. It's got a jaw like a snake with big fangs, like big. What are the big fat snakes?
Like
Anyway,
a viper or
not a viper,
cobra,
like
or
no, the big ones that are round and
Boa
like a
constrictor.
boa kind of.
like anaconda. Sure.
Anaconda, yes,
Yeah.
got an anaconda because he's got.
So
anyway.
boas and anacondas are constrictors, so they have they're tinier rear fangs. They don't have the big venom injecting fangs in the front.
Yes.
Anyway.
Yeah. So they had a big jaw and these kinds of large snake-like fangs from an older snakiness, but it also had little tiny legs that were very lizardy and gecko-ish. So it was kind of this, it had like old lizard traits, reptile traits, but also snaky traits. And so they were like, who are you? And so they gave it a new group. And so Brugnathar elgolensis has been added to a new group called the Parvoraptors. And these Parvoraptorids are called raptorids because they are thought to have hunted prey similar to raptors, like the dinosaur species, velociraptors. But the Parvoraptors puts them within this squamate group, apparently, because of the way the words work. So anyway, the clock for this particular find takes us back to the Middle Jurassic, 167 million years ago in Scotland. And it's a really great, almost complete fossil find. And because of the way they were able to see all of these traits, they're like, this isn't here in more modern groups. This is from older groups. And they've been able to look at these snake-like features, the teeth and jaws, and then also with the gecko, leggy features. And there are comparisons in the phylogeny that suggest that it could be more on the snake side, or in a different phylogenetic analysis said, no, it's more on the gecko side. And so it's a really interesting thing because it's still not answering the question of where snakes came from, but because of the dating and the particular traits that they are seeing, they are saying that this particular group of these parvoraptorids are taking us back to a very, very old age within the Jurassic. Co-occurring with dinosaurs, raptors, and others. So these squamates, the early where the snakes and the lizards come from. Anyway, there's traits. They have this is a great fossil find. They're really excited about it because it potentially takes the question of the origin of snakes versus reptiles or lizards, and it's getting at is this convergent evolution or is it actually origination? And so that is the question.
So sorry, where on this diagram is this new guy?
So for the phylogeny here, the stars, we've got a couple of stars, and depending on the phylogenetic comparison that they did, they put this dude in different groups. So either
that's extremely confusing.
Yeah, it's super confusing. They're like, oh, we compared it with this one data set and we compared it with this other data set, and depending on who's included and what data's in there, this dude could be kind of anywhere. So it's a mystery wrapped.
So this is interesting because I have learned just from kind of learning more and more about the evolutionary history of reptiles in particular, that reptiles are a junk group. There's no such thing as a reptile. It's polyphyletic. Right. It's a mess. But evolutionary time has shown us that a lizard-looking thing is a successful body plan. A lizard-looking thing keeps happening. Right. It's something that evolution returns to over and over again. And so it's understandable that, when people were first making their buckets, they were like, you're a reptile and you're a reptile and you're a reptile and you're a reptile, right? Yeah. But of course, like, crocodiles are more closely related to birds than they are to lizards. And then there's the tuatara, which is what I really wanted to see on the phylogeny you just showed. Rhynchocephalia is called out on there. So that's the tuatara, which is not a lizard and is the most lizard-looking non-lizard you've ever seen. But based on looking at their skull and the arches in their skull, that they are completely unrelated, or at least they're not a modern-day lizard. They came about in the Triassic. And so this is a very similar situation where they're like, you don't quite fit into any of these categories, but you look more like this.
However,
yeah, and this is not genetic. This is all morphological, and
that's limiting.
And so it's limited.
However, this is older than snakes.
Well, it's going back. So the question is the stem group, right?
Go back to that horrible graphic. Go back to the horrible graphic.
At what point in time did snakes and lizards splinter? Right? At what point in time was there that differentiation?
Yeah,
and there's still a massive question about this.
Hang on, no. There's a point in time when snakes have limbs. There's a point in time when snakes had, walking around on 4 little feet. We don't know what they look like. Maybe they were gecko feet. They were likely not hooved, in any case, right? And so if we're looking at this, it looks like it's pinpointing itself before the advent of snakes time-wise.
There's three stars, Justin. There's 3 different places that they think it might have landed.
Right. And these all look like they're somewhat before the advent of the serpentines on this list, or our known history of this.
One is a branch off of serpentes.
Yeah.
Right. Serpentines. One would be very closely related and and kept the legs, right? And then there's others that are building off of other forms, because again, this lizard form, like you're saying, is so conserved, that there's lots of versions that kind of look like somewhere in between. But in any case, what we could be looking at is a form of what a snake might have looked like before they lost their limbs. And I think that's a very fair way of analysis of looking at this creature. It's not necessarily the snake predecessor, but very much a picture of what the snake probably looked like before losing their limbs.
I love
think that would be rather fair.
the end of the abstract says, these findings indicate high levels of homoplasia and experimentation during the initial radiation of squamates and highlight the potential importance of convergent morphological transformations during deep evolutionary divergences. So they covered all their bases.
That's what I
there.
said.
They said, we don't know. Yeah, that's exactly what you said. I know.
Yeah, but all
I
of
said
these,
it,
all
but
these,
not as smart.
all of their stars are showing something that is in the ancestor pool
Yes.
of
And
snakes.
hey, they're all from Scotland. No, I don't know. I'm going to just say very, very quickly that I'm done with that story. Everybody, thank you for joining us tonight and enjoying this adventure through Science Land and the internet. And I hope that you are just having fun with us as we are having fun here with you. If you enjoy TWIS, please share with someone today. Make sure you head to twis.org. Help people subscribe to our podcast, to our live streams, to our channels, YouTube, Twitch, Facebook. And Patreon is where you can support us in an ongoing fashion in a way that, helps us not be able to fix the internet issues that plagued us this evening, but, hopefully buy us new microphones when we need them and keep our cables working well. So glad that you're all here. Thank you for your support. All right, coming on back. It is time right now for that part of the show. What? That part of the show. We call it Blair's Animal Corner.
With Blair.
Yeah, and I'm going to hit the music button again soon, but I realized that I didn't set everything up the same way that I did from the last one. So here it goes.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
Oh, goodness. I think I'm going to start with some crabs.
Lady crabs.
So this is...
Blair's got crabs, everybody.
Yeah.
Crabs.
Was this study from the University of Portsmouth Institute of Marine Science, and it is looking at crabs and their sensitivity to electromagnetic fields. Now, this is not some like crystals and incense situation. Underwater power cables actually emit electromagnetic fields, and there's generally an expectation that it doesn't do much. They're pretty low amounts of energy that are releasing from these power cables, but millions of crabs migrate along coastlines every year to reproduce. And so they are walking directly over, through, and around these electromagnetic fields. Now, just, as a Californian, and also as someone that, used to work at an aquarium on Pier 39, which is a crabbing, very heavily influenced kind of crabbing zone, I knew right away, like, oh, well, that's interesting because the take of crabs is very carefully monitored because crab mating seasons and the time when they lay their eggs is very specific. And so, in fact, you only can take male crabs. You are not allowed to take female crabs. If you have female crabs in your crab traps, you put them back. And there are specific seasons because you don't want to take crabs during mating season. You don't want to accidentally catch a female that is egg bound. And so there's lots of protections to make sure that the crab fisheries do not, collapse. Lots of people depend on crabs, and also a lot of animals, of course. Not to mention the fact that crabs are detritivores, and so they're like the janitors of the ocean also. So pretty gosh darn important ecologically. Well, what researchers found was that crabs do have a sensitivity to electromagnetic fields being emitted by these power cables, but specifically only female crabs have sensitivity.
Why?
Yeah, so they grabbed some crabs, popped them in the lab, 120 juvenile common shore crabs, and they exposed them to electromagnetic fields, covering kind of the intensities that you would find in underwater power cables. They started very, very low, and then they kind of ratcheted up to what you'd find in the high end. They showed significant behavioral differences between male and female responses. And of course, my assumption would be, oh, they hated it, right? No, female crabs were attracted to it. They were twice as likely to stay near areas with electromagnetic fields compared to those without them. So this is an issue not of hurting them, not of repelling them, but of attracting them so heavily that they don't move and disperse.
So they stick around the cable in the electrical field and don't go being excited by actual crabs.
Yeah. Males, meanwhile, not affected, no consistent spatial preference at all across any strength of electromagnetic field. They don't care. Yeah, and so they did, like I said, expose them to a wide variety of kind of levels of electromagnetic field, and they did find a statistical significant change in behavior for females, even at the lowest amount and also at the highest amount. So it was just across the whole span, they found responses from these female crabs. They did 10-minute trials, they tracked their behavior, they looked at zone preference, mobility, all this kind of stuff, and they used some tracking software. So they found that the adult female crabs, their movement was reduced by more than a third, 38%. And this was at moderate field strengths, which is about 1,000 micro taus? Electric units.
Teslas? No.
But they also found even noticeable effects at super low. And so I was just like, what kind of electromagnetic fields impact people? And so it depends what the source of the field is. It depends what the type of emitting is. But generally with electromagnetic fields, the main concern with human tissue is heating it. And for there to be any impact, it actually has to be like at least 10 factors higher than any of these highest ones that they were testing on the crabs. So
that makes sense. That's for like heating your tissue. That's like actually,
yes,
but not like
for damage wise, it's not.
But not your sensitive electrochemical nervous system responding.
So I don't want to. I know this was a study in crabs, but have you guys noticed there's a lot more electricity in big cities? And there's a lot more people.
Oh my gosh! I'm going to ask though. Like we do know that sharks and many fishes have lateral line systems. That the electromagnetic sensory system is very important. And it's interesting that like sharks that are predators, what other predator species are tuned in to that electromagnetic sense? And then why just the females? Have we ever looked to see if males emit anything in that electromagnetic spectrum?
Yeah. I mean, so it's there were a few things that kind of struck me about this study. So one is it's a very good reminder that when we put things in the ocean, we like to assume it's just like, don't worry about it. It's over there. It's underwater. You can't see it. It's all good. So there's that. There's definitely, it's, whether it's intentional or not, there's a little bit of that. But also underwater cables. So they're currently less than 0.1% of the ocean floor. But
they're all covered in female crabs.
But it's important to place them in the right place. If you are placing them along movement pathways of crabs, is that going to be a problem? Are you placing them in a shark nursery? right? Where are they getting placed? Because often these underwater cables, more and more of them are going in as part of offshore wind farms. And so specifically, this is part of the effort to move towards renewable energy, which is, of course, important. But when we do that, we need to make sure that we're also paying attention to, how we're impacting. But the other piece of this that I think is a good reminder is the fact that the males and females had such drastically different responses. So it is another reminder that you cannot study literally anything on any animal subject without testing both males and females, because you have no idea. No one could have predicted that male and female crabs care more or less about electromagnetic fields. Like, that is so unexpected and such an excellent reminder that you have to test both. And especially in this case with crabs, you don't want to pull the females from the wild because of fishery rules. So, like, do you get your extra permits? Do you pull females? Or do you go like, oh, I don't want to make that ecological impact. I'm only going to pull males for my study. Well, then you think that electromagnetic fields don't matter. So it's still really important to do that work.
I immediately have like 3 questions.
Yes.
One is in the chat room. How do you
tell them apart? Yes.
Yeah,
so
male
it is.
Female crab.
So if anyone wants to Google image search male versus female crab, I'm used to looking at it in Dungeness crabs, but this is true in other crabs too. Think about a crab is a squished lobster, right? So a lobster is a tail. The crab has a tail also, but it is bent under and flush with the body. If you pull the tail off, it looks like it has a perfectly fit kind of like indentation that it lives in, right? Mm-hmm. And so the tail is a completely different shape for males and females. I think the female is super wide. It looks like almost like a circle. And then the male, it looks like more like a pointy triangle. I think that's the difference. The male is the pointy and the female is wide because all the eggs are hidden under the tail.
Yeah. Okay. So you just look at the tail itself.
It's very obvious once you know what you're looking for. Yeah.
Okay. Next question. The next thing that occurred to me is, could this be used as just the new habitat? Is it perfectly acceptable for them to be in this location? Will the males go and find them? Can this just be like, this is now where crabs live? And that's fine.
So you want dispersal. You don't want all the crabs in one place.
You don't want to put all your crabs in one crab basket. Okay.
Yes, that's
And
correct.
then the next one I was thinking is, does this have anything to do with crab preference? Or is it a crab preference? Like the crabs are detecting it, right? The female crabs are detecting something in the electrical. But why?
Mm-hmm.
It's probably not a preference for an electrical field.
Right.
It's probably a signal for something else, something that they would maybe a nutritional factor, something that they would eat that's normally itself attracted to or generating.
Yeah. So Kiki brought up a good point about that. With sharks, for example, their lateral line detects electrical signals because of like heartbeat, basically. So they're sensing the chemical processes of life. So like they're sensing the electrical current of life.
Yeah.
So that could be it. Absolutely. It could be that crabs are like, ooh, little fish I can eat. Krill, right? Like totally, that's possible. But they are detritivores. They usually eat like poop and dead stuff. So it's I don't know. I'm not sure. I think that's a really good, rich future study is figuring out what it is about the electrical signal. Is it just something that's inadvertent that's triggering a chemical pathway in their body? Who knows?
My point would be to go look at existing crab fields and check for like where they tend to congregate and then just see if there is some sort of background electrical field being generated from something in there that they're already being attracted to, that we just maybe it's weaker, maybe it moves. And if it moves, is it moving with currents? Is it Kiki, we can't hear you. So
Sorry, but this is
unfortunately,
weaker and it's getting in the way, right? So it's
Mhm.
getting it's stopping it is attracting and disrupting the natural behaviors of these crabs, and that's
Mhm.
bad.
Mhm.
So Blair said, let's be smart.
Mm.
Like with the dodo.
Yeah, speaking of being smart, why are we bringing back the dodo? Um, we're not actually, but Colossal was in the news again, and I just wanted to bring it up to kind of tell everybody what's going on, because they were in the news again, and I wanted to talk about what actually happened. So, the headline was, Colossal makes huge leap in bringing the dodo back from extinction. What they did was, they cleared an early hurdle by growing primordial germ cells, which are the precursors to eggs and sperm, from the rock dove, aka pigeons. So that's it. They created pre-gametes. They did it.
But rock dove pre-gametes, not dodo. It's pigeon.
Yes, because
they're related, and yeah,
it's
right?
they want to have the primordial germ cells stable enough to be able to gene edit them at that stage, because with mammal reproduction you can edit like DNA in an egg. Okay. But you can't do that with birds.
Because of the shell and all the things.
Well, yeah, and because the egg is actually developed much later, like the fetus starts developing before there's an egg, basically, because the egg is like it's coinciding that the egg is developing as the fetus develops. And so because of that, they can't use the same gene editing techniques of how they would try to do it, like with the southern white rhino and the northern white rhino egg. Like they can't do it exactly the same, right?
Or like they reported on the dire wolf, right?
Exactly. So they did the dire wolf thing. Yeah.
Wait, hang
It's
on.
still not real, but yeah.
So this actually could be a major. Are you saying this is a major breakthrough for bird conservation?
Pigeons.
It's a pre-breakthrough. Because all they did was pigeons. All they did was make pre-eggs out of pigeon. I would argue the big jump in bird conservation would be editing that pre-egg. But they haven't done that yet. That's actually the thing that's going to get you anything. Right now, they're just like, we almost made an egg. Is basically all they did. Which, that's not even their plan. They're not going to do the rock dove, the common pigeon. They actually have to do the Nicobar pigeon in Texas, which they have established a breeding colony of for this activity. That's the one that they're going to have to do, because they are saying that is like the closest living relative to the dodo that they have, is this Nicobar pigeon in Texas. So they have their germ cells for editing. Then they're going to be able to edit what they have in the Nicobar pigeon germ cells, and then put them into chicken embryos. And then the chickens would grow up with pigeon making cells in their ovaries or testes. They would be the surrogates. They would lay an egg with a dodo in it.
With an organism that is an approximation of the genetics, the genetic representation of a dodo.
Correct. So it's actually, it's 3 different birds. Yeah. But one of them is a guess, basically, because we don't have a dodo genome. So they're basically just like, they're going to fiddle with the chickens and the pigeons until they get something that looks like a dodo, is what they're trying to do.
Why don't we have a dodo genome?
We do, that's why but they have to mess with the pigeon genes to be like, they have to get it going so they can edit the pigeon and chicken stuff to be able to be like, we're putting in Dodo genes. And
so we have a pigeon genome, but it's not, you can't just inject that into, you have to make it functional with the pigeon genome. So you can't just go, I'm going to put this in here. Exactly. So you're going to have your
new generation, and then you have to do it again to add more Dodo gene in. So it's going to be a fast track of genetic evolution in a lab.
But to Justin's point, Colossal's chief science officer says, by developing these protocols, we're establishing crucial biobanking capabilities and opening new possibilities for genetic rescue of endangered species. So as we've said before, this like PR nightmare is really just fueling their ability to do just groundbreaking conservation efforts later. All that to say that they have a place to put these dodos, which I think is bonkers. They're going to put them on an island country off the coast of East Africa where birds were endemic before they went extinct. So they like identified habitat for the dodo already, which I think is insane. But I just wanted to bring to daylight that once again, I mean, this is
that's. To be fair, you like if they hadn't identified at least a place where they would plan on putting them, you'd be like, and they don't even know where they're going to put them. Like,
let's be real. They are not going to make a functional. They're not going to make a functional population of dodo. That is not going to happen. They're going to have their 20 dodo that they have in zoos and they parade out at the bioscience conference on a leash. Like that is what's going to happen with these animals. They are not going to reassimilate into the wild. That's not how, let's be real. That is not what is going to happen here.
Okay. But the technology they develop could return a variety of pigeon, perhaps, back to the wild.
Sure.
After it became accidentally
I am not
extinct.
saying it's not like sound science, but I think, as is always the case with these colossal articles.
Yeah.
I want to, put some daylight on this and say they are not bringing the dodo back from extinction. Even if they made something that looked like a dodo, it wouldn't be the dodo that they were bringing back from extinction, just like it wasn't a dire wolf that they brought back a couple months ago. It is not. It is an approximation.
That one hurt my heart a little bit.
And so they're not doing that. They're also, like I said, they're not going to bring these guys back to the wild. They're not. This is all proof of concept that's like sexy and exciting for people to talk about in the news. Cause like, the dodo, I would love to pet a dodo. Oh my God. I'd love to feed a dodo a worm. Like it would bring so much joy to my heart, but it is also important that we remain grounded and recognize what's actually happening.
Sensationalism, PR in the service of science.
And we'll be damned if we give them any publicity here.
Yeah, they're gonna have.
Even though I've said their name 20 times.
Many patents. Oh my gosh.
Yeah.
Wow.
Hey, one bright spot.
I'm gonna tell a story.
By mixing it with the chickens, I had always heard that dodos tasted terrible. So maybe
now they'll taste like chicken.
They must have tasted great.
No, my understanding was it was just a matter of they were easy to catch. Because they weren't afraid of humans, but that they actually it's just sailors stuck on an island,
Oh, tough and greasy.
Yeah,
oh, sure.
Apparently
but you better believe Charles Darwin ate one. He ate
now
everything.
They will taste like chicken. Justin, you want to talk about something techie?
Yeah, I brought a tech story today, which is I've never heard of this. So everybody needs to Google the G1 humanoid robots.
Whoa. What?
This is that human, okay, I'll just read the story because I'm not hip to the fact that we've got humanoid robots running around, but it says,
oh no, it's this one. I hate this one. All right, I'm sorry. I just looked at it.
Researchers have, it looks like a Cylon. Like the ones without the skin
except
on,
black. Right? Like the black, yeah. When they were silver and black later.
Yeah.
Okay, yes. Go ahead. Sorry.
Researchers have uncovered serious security flaws with the Unitree G1 humanoid robot. something that's already being used apparently in laboratories and in some police departments.
What?
That's what this story is saying. I believe every word I hear, but I don't. Oh my gosh. They discovered that G1 can be used for covert surveillance and could potentially launch a full-scale cyber attack on networks. So this is all
all within China, right?
Sounds like this stuff of science fiction nightmares, robots that are secretly spying on you and could be controlled by remote hackers. However, the concern is real as these types of robots are becoming increasingly common in homes, businesses, critical infrastructure, and public spaces. So this is the article is by Paul Arnold over at phys.org. It says, in a new study, cybersecurity experts from Alias Robotics describe how they performed a digital audit on G1, reverse engineering its internal software and eavesdropping on its internal communications to identify critical weaknesses. One of the most serious flaws was its Bluetooth low energy setup for connecting to Wi-Fi, something that's apparently common to many consumer robots. So they found that the encryption protecting this process was incredibly weak and easily broken as it relies on a single secret digital key hidden inside every Unitree robot. And simply encrypting the word Unitree with a hard-coded key was enough to bypass security and gain control of the robot's entire system, which means hackers could easily take it over, inject some malicious commands to crash it or make it attack other devices. Equally concerning was that G1 acts as a Trojan horse, secretly and continuously sending data to servers in China every 5 minutes without any sort of acknowledgement to the user that this is going on. Team showed that G1's onboard computer could be repurposed for offensive operations. Additionally, robot's custom encryption method to protect its internal configuration files is fundamentally flawed because, again, simple static key that's the same on every single robot. So if you figure out how to unlock one, you got them all.
Oh my god. So this is the other thing. I just looked these up. They are not as expensive as I thought they would be.
No? You want one now?
No, absolutely not. Never in a million years. But no, I mean more that just like if they're so easy to hack, but they're relatively cheap. They're like cheaper than a car. Like just I think that compounds the issue a little bit. That, you know, if they have their way, there'd probably be quite a few of these running around, literally.
It seems though like this is a basic technology issue, right? That networked devices, we have the back doors, we have like issues related to the DJI drones across the country. Like they just reassessed and said, nope, they got back doors. You're probably giving data to China, so people still aren't allowed to use their DJI drone technology. But these kinds of devices, if they're being put all over the place in sometimes, you know, high security or in, you know, areas where
can't trust a human with the job. Oh, we'll get a robot. It can never betray us.
It's crazy. Like it would take so much work for a human to like record something or like copy something and share it.
Send it to their secret contact in China.
But meanwhile, you have the robot. It's just like, oops. Just it's
every
open.
5 minutes. Every 5 minutes. That's wild.
Oh.
Wow. And you know, this is unrelated, but I'm just gonna quickly throw out there. Can we stop making robots look like humans? They don't need to be bipedal. They don't need to look like that. They don't need to have a head. Can we stop? Can we, and especially until we figure out how to not hack them, can we stop?
Like headless robots. Okay, everybody.
I want something with like a tank tread situation, like more like Rosie from the Jetsons. Can we do that instead? Could we just move towards just like the arm on a tank tread? It's not make it look like a human. That would be my
Well,
preference.
I don't mind them making it look like a human as long as they make it out of cheap plastic, right? If the future robots are made out of some kind of cheap lightweight plastic, so if they do turn into murder bots, you know, and they try to attack you, they got like
Just
no
give them a nice,
limb strength.
like, little swipe, trip them. Because their
just
bipedal design is really dumb, actually.
Yeah.
Cheap beach chairs from, you know, the variety store.
Exactly. Don't give me the Rosie the tank. Give me some cheap plastic robots that are easy to push around, and I think, you know, I'm less scared of them being murder bots. But now that I know that they're betraying us, our every secret, and the other thing is like that idea of the connectivity in today, like where things are like connected to your central computer, you can turn your lights and stereo up and down, and everything's all connected. And you put this robot,
smart homes,
in that smart home network, and then suddenly it's compromisable and can turn everybody's lights off at once, or disconnect your refrigerator, or order stuff on Amazon for you. Like, it becomes a problem.
The problems you just listed were ridiculous. Oh no! Bad networked robot turned off my lights.
No, everybody's lights. Everybody who's connected to, like, across the nation. Like, you got to understand, this is like in every. It's like a really common Doctor Who theme, where like something that becomes ubiquitous within society that everybody's like, oh, this is a normal thing, and then you find out there's like some sinister alien plot behind all of it all along, and then turns on everybody.
And I totally agree.
Step one.
There is the TV
Step
show,
two, murder bot mode.
It is one of the best book series. I love this book series so much. Murderbot Diaries, highly recommend if you have not read them. Very fun if you like murder bots. Yeah, it's great. It's good. You know, imagine blood. It's fantastic. I have two stories that I will get to over a very long time because it's early still. No. Think of this show like a super centenarian right now. The length of the show is like the lifespan of the world's oldest people. What is it that defines the longevity of those individuals? And what is different about those individuals from the people who just live normal lifespans or shorter lifespans? So researchers just published in Cell Reports Medicine their analysis of one individual compared against a whole bunch of other people. So they did a complete multi-omic blueprint of a super centenarian, female aged 117 years old and 168 days. Caucasian woman born March 4th, 1907 in San Francisco from Spanish parents and settled in Spain since she was 8. She had a lot of great life experiences. She lived in Catalonia. There's another place, M116, another long-lived individual. Life expectancy in Catalonia for women is 86 years. So that's exceeded by more than 30 years. So what's going on there? And so these researchers decided to look at everything. All of the genes, all of the proteins, all of the transcriptome, so RNA, DNA, proteins, microbes, everything. They looked at it all. And what they found is that the majority of the features of this individual were those of a younger person. Immune response, microbiome, epigenetic clock, the inflammation, cardio, metabolics, all these things were similar to someone who had not really gotten into the downward slide of aging yet. Yeah, hi, what do you want?
Well, I don't mean to interrupt the beginning here, but when you say
do.
younger person, I did mean to. I
Yes,
raised my hand and I
you
started talking.
did. No,
When they say younger person, I'm wondering if they mean an 86-year-old,
no.
right? Or somebody who like younger than 117 leaves like everybody.
Right, it does. Absolutely. Okay, so the comparison that they did, they were really looking, they compared this one individual to like massive database of multi-omics of individuals. And so the comparison that they found is the chromosomal clock. They found that the telomeres, the short telomeres, they had a smaller number of short telomeres. They did find, and they say, actually, no, I said that wrong, and I'm sorry, I'll correct myself right now. We talk about telomeres being the ends of the DNA, the protective bits that get worn down as cells divide and that protect our DNA. This individual had a huge erosion of telomere sequences. So what's going on there? So is the telomere attrition more of a chromosomal clock rather than a predictor of the diseases or the aging that's going to come?
Wow.
So it's a different way of looking at telomeres.
That would be wild if we kind of completely shifted our thinking on telomeres because of 117-year-olds.
They're involved, but but let's reframe it, right?
I'm glad that that's there because it's evidence that this person is of the age. I'm always suspicious.
They are old.
Well, it's because like they have to have, started life in a time with really poor record keeping. And
yeah, and you reported on the blue zones and how those were all kind of poor record keeping. Yeah.
Well, and then like all the 20-something-year-olds in China come from a region that only started tracking births in like the 1950s. So like they had like makety up birth certificate times or ages, right? They were inventing their own birthdays once people started coming around and creating IDs. Like they didn't even have IDs at that point. So then I'm like, San Francisco, Great Fire, records can't have been there. Oh, maybe this family fled to Spain after a murder and it was like, oh, our name is this and this is our child so-and-so. And they're like, oh yeah, she's really a child. She just looks, whatever the thing is, right? Like, I'm always suspicious of age. Now hearing this highly deteriorated telomeres that's tracking with a 117-year-old makes more sense to me than them just having younger features, because then I'm like, they're just not old.
Right. So they are older, right? They are older, but they've got aspects that are similar to younger individuals. And you asked about the age, and the younger individuals are around 25 years old.
Wow. That's incredible.
Yeah. So these individuals who are living 110 plus, right? These individuals really, they are younger forever. They're not aging the same way that everyone else does. So this woman, she did still have age-associated B cells. There were immune cells that were related to aging and being older. There were issues related to blood volume, hematopoiesis. So the ability of and how your blood volume is being turned over. So there's stuff that was getting older, right? And it was not super young anymore. And there were mutations, which is what you expect. As you get older, as you live longer, you accumulate more mutations. So there should have been more mutations found in certain places in this analysis, and they found them. But it's really very interesting. There's the good living aspect of it. I mean, Catalonia, probably not a terrible place to live when you're, probably was great. Mediterranean diet. I don't know. She lived through a couple of world wars. She lived through flus. She lived through massive pandemics, global flu pandemics. She lived through a lot of things. And still was younger
Live
than
to
me.
tell the tale. Yeah.
So the lack of, or the low grade inflammation, the lack of inflammation is really interesting. My guess, I'm going to put out a guess that this is going to come down almost 100% to mitochondria.
Possibly, right? Or at least, the genetic profile or the immune system that allows the mitochondria to stay healthy. Right?
Before that, it's going to be before that. It's going to come down to the ATP and the, what is it? But
also getting the trash.
The nucleotides. Within the mitochondria build up. The precursors for RNA tend to build up over time. And this is where you start to see senescence of cells and the, what is it called? That signaling cascade that sets off the alerts that starts inflammation. That whole process begins in mitochondria and has a lot to do with whether or not it has accumulated too much precursor, because mitochondria doesn't normally have a way of getting rid of this. Now, her mitochondria might be able to balance itself and expel these precursor RNAs so that they don't become burdened, so that balance stays good. And so those mitochondria, we got to study it and figure out how she's getting rid of it, because that could be the key to unlocking the fountain of youth.
But you're guessing at this when this is an entire systemic analysis, and
it looks like it.
The mitochondria are very, very, very important. I mean, really, truly.
But I'm gonna predict that that's where this is gonna end up, because this is not part of the study, but there have been these studies that have looked into the building block nucleotide buildup in mitochondria's effects on inflammation and aging of cells. Like the main things that we see as the aging of cells begins with and maybe even ends with mitochondrial health.
But if the
mitochondria,
immune system and the garbage, the waste system is working fine, then the mitochondria can stay healthy. I mean, that's one of the aspects.
But they still age. I mean, that's where aging happens. That's where the senescent cells happening. It's all mitochondrial health that affects cell health, that affects tissue health, that it expands from there.
Yeah, I mean, she was old and she died.
Because I needed to study her mitochondria.
Aging happened. This wasn't a mitochondria study. This was just looking at this person, and I like your hypothesis.
That's why I'm like, how is that not? So this is after the fact that she's gone, which is unfortunate. They need to find those super agers now and do just a complete mitochondria study on them to see if it is. Woo.
I think that is that could be very interesting. I was pointing up to where Blair is in my thing, because look at her face. She's so tired. I'm trying to finish.
I'm fading fast.
I know. I have one last story, and that's it. That's it, everybody. One more story. Maybe if you want to have good memory, don't eat junk food. That's it.
What'd you say?
No. Did you forget already? Oh my gosh. What'd you have for dinner?
Cookies?
Yeah, researchers at UNC School of Medicine have just published a study in mice, not in people, but in mice, because they're looking at mouse metabolism. But oh my gosh, when they gave the mice high-fat diets, that were supposed to be like our highly processed sugar fat diets of the West, they found that it led to a change in the way that the neurons used glucose, and they became less likely to do the job that they were supposed to do. And so neurons in the brain, the interneurons in the hippocampus, which is part of the memory hub of the brain, they they got weirdly active, but they were not modulated in the way that they should be by the amounts of glucose that were present. And so what they are suggesting is that there are very sensitive memory circuits. They're really sensitive to what we eat, and that this is not related to how much you weigh or whether or not you have a metabolic disorder like diabetes. This is specifically just brain glucose levels, fat in the diet, and the response of these neurons to what they are saying is, mouse junk food. They did find, however, even though the neurons got all messed up and made it so that basically short-term memory and all that stuff is not going to be great after you eat junk food. Hey, college students, like cramming for your finals, junk food during that period, maybe, eat a nice meal, go to sleep. Also, intermittent fasting, they found reversed the effect of the junk food. So the junk food messes things up, but when you go, let's start over with some intermittent fasting, and maybe there's other stuff you can fix it. If you eat junk food, all is not lost. Your memory will not be completely gone. But that fits also. I hope you like your cookies.
Also fits an energy use strategy, meaning we have a lot of energy now. Let's not waste any time remembering what's going on. Let's go hunt. Let's go fish. Let's go do whatever a mouse does with its time off. Let's go get more resources. And now that we're fasting, now we can think back on life and ruminate about things for a minute.
But if you think of the way that animals eat, and then normally you don't have junk food, right? Like maybe you are a shark, a great white shark, who gets to eat the seal candy bar, right? But with all the high fat and everything in the blubber. But more often than not, what you're dealing with is an evolutionary system that is set up to rest and digest after you've taken on a very high calorie fat meal, not keep going and act like that was lunch. 15 minutes standing up or walking on your way from place to place. So the way that we function is not the way that evolution or adaptation led us to where we are. Anyway, junk food.
Can you write me a note for an extra long lunch break at my work?
Yeah.
Blair needs to process her food or her memory will be bad.
Yeah, if you want her to remember anything in the afternoon, you really need to make sure she has time to eat good food and
a 2-hour lunch.
Yes. Lunch, exactly. Anyway, might just be telomeres and mitochondria. I don't know. I hope everyone will remember this show forever. I hope you have all of the stories and the conversations that we have been having just in your brain, because this isn't junk food. This show is nutrition. Are we done? Ready to go?
Let's do it. Let's close it out.
That's what I was gonna do. Justin looked like he was gonna fight it for a second. I'm like, what?
No, I'm ready. I gotta be at work in a couple of hours.
I know. Thank you for listening, everybody. Thank you for being here. I'm so glad that you joined us for this extra long show that we were going to finish at 10 o'clock. Shout outs. Fada, thank you so much for your help with show notes, social media, everything that you do. Identity 4, thank you for recording the show. I know tonight's going to be a little bit weird. Gord, Arnlor, everyone who's in the chat rooms, thank you for keeping it wonderful places to be. Thank you for enjoying chatting and being part of it all. Rachel, thank you for editing the show and assisting in the way that you do. Thank you to our Patreon sponsors. Can never forget them. Hey, everybody. Thank you to Aaron Anathema, Adam Mishkan, Ali Viola, Ali Coffin, Andrew Swanson, Arthur Kepler, Artyom, Bob Calder, Bob Coles, Brendan Minish, Brian Carrington, Chris Wozniak, Christopher Dreyer, Christopher Rappin, Craig Landon, Craig Potts, Dana Lewis, Daryl Myshak, Dave Wilkinson, David E. Youngblood, Don Mundus, Donovan Styles, a.k.a. Don Stylo, E.O., Eden Mundell, Eric Knapp, Flying Out, Fredis104, G. Burton Lattimore, George Corris, Greg Briggs, Jack, Jason Olds, John Atwood, John McKee, John Ratnaswamy, Johnny Gridley, Ken Hayes, Ken Northcote, Kevin Parachan, Kevin Reardon, Kurt Larson, Lawn Makes, Lauren Gifford, Mary Gertz, Marjorie, Mark Hessenflow, Noodles, Patrick Pecoraro, I hope you're doing okay, Paul D. Disney, Paul Ronovich, Philip Shane, Pierre Velazar, Pete I.G., Richard Badge, Robert Norland, Robert Farley, Rodney Lewis, Rhea Garcia, Remy Day, Sean Laris Niam, Shubru, Stephen Alberon, Steve Leisman, aka Zima, Sue Doster, Teresa Smith, Tony Steele, Vagard Chefstad. Thank you, all of you, for supporting TWIS. We really cannot do this without you. Your support is essential. And if you want to support us on Patreon, head over to twis.org and click on that Patreon link on next week's show.
We will be back. Well, some of us. Wednesday, 8 p.m. Pacific Time, broadcasting live from our Twitch, YouTube, and Facebook channels.
Hey, do you want to listen to us as a podcast? Perhaps while you drive to pick up your fast food dinner that you probably shouldn't have for your brain? Just search for This Week in Science or where podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you've heard here, today's show notes, links to stories will be available on our website, www.twis.org, where you can also sign up for Blair's newsletter.
We love your feedback. If there's a topic you would like us to cover or address, or a suggestion for an interview, please let us know in one of our social media accounts, or better yet, send us an email. Just be sure to put TWIS, TWIS, on the subject line, or your email will have no ties to a phylogenetic tree. It'll just be a bunch of dotted lines, and we'll never see it at all.
Yes, but we do look forward to discussing science with you again next week. And if you've learned anything from the show, please remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in 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 Science.